All right, hi everybody. As usual, I'm just going to wait 1 second, make sure that the sound is coming through and everything looks good, and then we'll be off to the races. Okay, everything looks good. So, as usual, anybody have any problems on the audio-visual side, just let me know, and I'll do my best to make sure everything's working. I see David Neff is here. Thank you so much for your help on the audio-visual side last night. I appreciate it, and I will never screw that up again, so thank you. So tonight, we are doing part 3 in our series on evolution for creationists. This is to remind people, if you are new to this series, this is basically an introductory evolutionary biology series. So we're going to just go through the basics to lay a foundation so that anyone participating in the evolution creationism conversation is all on the same page as far as the basic terminology, the basic concepts, that kind of stuff. Now, we are now getting into our unit, if you want to call it that, on genetics here. So we're going to start tonight with just a little bit of kind of how the field of genetics got started. We're going to keep going with that next time, and then we'll have at least a couple more nights on evolution, on genetics, I should say, kind of basic genetics after that. So it's looking like today we're going to do Mendel just a little bit. Next time we'll also do Mendel. And then after that, we're going to get into Thomas Morgan and chromosomes and that kind of stuff. And then after that, we'll do a little bit of population genetics and Hardy-Weinberg's kind of stuff. So basically, if I keep this up at once a week, no promises, but that's the plan, then we're looking at about 4 weeks of basic genetics. And then after that, I think maybe we'll do natural selection, which will be 2, maybe 3 episodes. So we're going to see. But for right now, we are going to start our unit on genetics, and I figured out how to make it so you could see my cursor, so that's nice. So this will be a lot of fun. So as always, we're starting with our outcomes here. What do I want the viewers to be able to do with what we talk about today? I want you to be able to compare and contrast the competing hypotheses for how inheritance works. We're going to talk about 2. We're going to introduce 2, and then we'll really talk about 1, and then we'll cover the next one next time. I want you to sequence what Mendel did, so like what were his experiments, and then evaluate how his work disproved one of those 2 competing hypotheses, the blending hypothesis. So be able to look at his data and tell me, what does it mean and how does it disprove that hypothesis? So now that we've got our outcomes out of the way, let's talk vocabulary, because now that we're into the genetics, there are a bunch of terms we all need. And my assumption here is that nobody knows anything. My assumption is 0. So we're going to build this from the ground up. So we need to start with just some basic terminology. So here, first, most basic term in genetics is the word where the term genetics comes from. It's gene, which is just a unit of hereditary information. It's a unit of heredity that you inherit from your parents. An allele refers to an alternative version of a gene. So the simplest way to think of an allele is for something like hair color. Now it's more complicated than the Mendelian system we're going to talk about over the next couple of weeks, but obviously people have different colors hair, and that has to do with different alleles for the genes that result in the pigmentation for hair. So you can have alleles for red hair, alleles for brown, black, blonde, lots of kind of variation in between. So alleles are just variants of genes. We can also talk more broadly about characters and traits or character states. So a character refers to an observable heritable feature of an organism, and a trait or a character state refers to the detectable variant of that character. So to put another way, for like sticking with hair color would be a character, and then brown or red would be the trait or the character state. Okay, there's kind of the distinction between character and trait. And then finally, we need to talk about genotype and phenotype. Okay, genotype just refers to the genetic makeup of an individual. Generally, when we talk genotype, we mean what alleles are present at a specific locus. But you could also talk about genotype in the kind of broader genome-wide kind of scale. And then phenotype just refers to the observable physical traits that an organism has. So your genotype is what alleles for hair color do you have. Your phenotype is what color is your hair. Okay, those are how those terms are related to each other. Before we get into Mendel himself, I just want to point out that you can basically group these together like this. You can think about genes are for characters, and then alleles are for specific traits. And then the genotype refers to which alleles are present, and the phenotype refers to which character states are present. So these terms, they're different, but the differences are kind of subtle and they're all related to each other. So just be aware of how these terms relate to each other. Uh-oh, we're buffering in and out. That's not good. Hang on, everybody. Let me just pull up my view of this and see what's going on here. Sorry, I'm just going over to YouTube to watch and see if there's a problem. I'm looking for buffering in here, and I'm not seeing it as I go back. Was there a specific part that cut out? Anybody else? Can everybody hear me okay? It seems to look okay on my end as I'm going back through. We okay? Okay, I'm going to keep going then. All right. Thank you for letting me know. Okay. All right. So back to, well, now we can actually get into Mendel. So we're gonna move on to the next thing. Okay. So now let's talk about Gregor Mendel. We need to get into like the background of what we're talking about before we get into the experiments. So the person we're talking about tonight is Gregor Mendel. He was an Austrian monk. He lived from 1822 to 1884, and he matters because he figured out the basic rules of inheritance for eukaryotes. And I want to be clear that we're talking about eukaryotes here. Bacterial genetics and prokaryotic genetics are different. We're not worrying about that here. We're just talking about eukaryotes. Now, he published in 1866, and it got very little attention. Basically disappeared for the better part of 30 years. Nobody really paid attention to it. But it was rediscovered in right around the turn of the century, right around 1900. And then people kind of picked it up and did further work on it. We'll talk about that in a few weeks. And this work formed the basis for the modern field of genetics. So Mendel is considered the father of genetics. Now, there is kind of an apocryphal story. I don't know if it's true, but the story is that when Darwin died, and Darwin also died in the 1880s, I forget the year, but when Darwin died, he actually had like a letter or something from Mendel on his desk, but he hadn't read it. So that would have been one of those great like aha moments if Darwin had read Mendel's work and to be able to put two and two together to see how the genetics underlying evolution work. But though they never actually connected and were able to put their ideas together, which is a shame. So now another character in the story that needs an introduction is the modest garden pea plant. This is Mendel's experimental model organism. And for those of you unfamiliar with the term model organism, that is simply an organism that you can use to study because it's easy to work with, that you can then take those findings and apply them to lots of other things. So like, for example, for eukaryotic genetics, yeast is a really good model. It's easy to grow. It has fast generation times. It's not dangerous. But all the processes work really the same way to like how they work in human cells because eukaryotic cells are very, very similar to each other. So Mendel was working with the garden pea, which is very cheap, easy to obtain, lots of traits, and we're going to see examples of that in a minute, easy to grow and have a relatively short generation time. And if I recall, I think it was about 6 weeks, I think is a generation time. So or maybe 8 weeks. It's relatively short. So that makes it easy to do a bunch of generations relatively fast. The other nice thing about the garden pea is that it's easy to control pollination. So you control who's mating with whom. So you have what Mendel would do is he would actually take a paintbrush and he would go and manually pollinate between the plants so that the matings that were occurring were the ones that he wanted, so he could cross the traits as he wanted to see how the offspring turned out. And also conveniently, there were many varieties available, so Mendel ultimately collected 34 strains of garden peas and then was able to mix and match the traits in different ways to see how the inheritance pattern looked. So now let's look at his experimental approach, which was also unique at the time. So Mendel was not just like mating things and seeing what happened, because some of the stuff that he would have observed is stuff that we knew. Offspring look like their parents. We'd been doing that since the agricultural revolution, like 12 or 15,000 years ago, right? We know how to like domesticate things and how to do selective breeding. The important thing is Mendel was counting. Right. So he was saying, okay, you have parents, X and Y, and you get kids and how many of each type of kid. He also documented his protocols meticulously. So if you wanted, you could go back and redo his experiments right now. We know exactly what he did. And the last thing he did that was super important was recording the data. Because remember, kids, the only difference between screwing around and science is writing it down. Now, the starting point for Mendel was something called true breeding lines. These are lines of pea plants that, for whatever trait he was looking at, whether it was flower color or seed shape or whatever, he had true breeding lines. This just means they always had the same trait. You could self-fertilize them and self-fertilize them and self-fertilize them, and they would always turn up the same. And in order to use a line of plants in his experiments, they had to be true breeding for the trait in question for 2 years to make sure there were no other whatever factors it is that's causing the inheritance of the traits. Is it buffering again? Why is it buffering? Everything, it looks good on my end. I don't know what to tell you all. Everything looks good on my end. Anybody else? Oh, I do see it buffering. That's unfortunate. That's weird because everything is the same as before. I haven't changed anything. Well, I'm just going to keep going. And sorry, everybody. Did you miss anything important? Did you miss anything important? I don't think you missed anything important. How long are the cuts when it's cutting out? How long is that? Is it like a couple seconds or is it like 30 seconds? Sorry, I'm just taking a second. Okay, see you later. Okay, so this idea of blending inheritance, this was the prevailing idea. The alternative idea was called the particulate inheritance hypothesis. So this is the idea that there are discrete heritable units carried by gametes. So instead of having fluids and they mix together, you've got discrete things. Buffering about 30 seconds. God, you know what? Okay. Hang on, everybody. How can I fix this? If I leave, then the stream ends, so I can't do that. I don't know what the problem is because my setup is exactly the same as it's been always. So, yeah, sorry. All right, tell me here. Hang on. I'm going to be in the chat because that's faster. All right, you know what? Don't tell me if it's buffering anymore. I'm just going to keep going. We're going to deal with it. Yeah, it's try removing yourself from the stream and adding back in. Hit remove from the screen. I can try that, but I don't want to remove from the stream because then I think that will end it if I'm myself back. I don't know. All right, just keep me appraised of stuff, but literally nothing has changed since ever on my setup, so I don't know why it's giving me problems tonight. Yeah, in fact, there's a lot less going on right now compared to last night when I had like various YouTube windows open at the same time. So we're just going to go. Okay. Particular inheritance hypothesis. This is the idea that traits are discrete units in the gametes rather than fluids that are mixed together. Under this hypothesis, we expect offspring to resemble one parent or the other. You could have intermediate phenotypes, but you don't have to. Whereas with blending, you need to have intermediate phenotypes. So let's talk about how we can test these two hypotheses. Now, Mendel used something called experimental crosses. This just means you're mating two organisms together to see the offspring's phenotype. These are going to be the true breeding lines that we talked about before. They're going to have opposite traits. So like you could have one that's tall and one that's short and see what the offspring look like. Are they tall or are they short? And if they're a mix, what are the ratios? So we're going to do these experimental crosses, document the offspring, and then use those results to evaluate our two hypotheses. So in these crosses, you have three generations that are relevant. You have your P generation. Those are your true breeding lines, and P stands for parental. You have your F1, which is your first filial generation. That's from the Latin for son. And then your F2, you could think of those as your grandkids. And what Mendel would do is he would have the P generation, then the F1, interbreed the F1s to get the F2s. And then you could evaluate your frequencies for the different traits and compare your hypotheses, compare your predictions. So here is your prediction. If blending is accurate, the F1 phenotype should be intermediate between the P phenotypes. So if you have parents, for example, that are red and white, and blending is the appropriate explanation for how this trait is inherited, then what are we going to expect in those flowers, in the offspring? They should be, I'm assuming everyone just said it when you heard it, they should be pink. Right, so that's what we'd expect with blending. Or if it's purple and white, you expect light purple. If it's tall and short, you expect intermediate height. You expect the offspring traits to be somewhere in the middle. And then in subsequent generations, you don't expect the P generation traits to reappear because once you mix them, you can't unmix. That's what we predict based on the blending hypothesis. So here's what Mendel did. We're going to start with true breeding P generation, okay? And for example, we could use flower color. So we're going to use purple flowers and white flowers. We're going to interbreed those. We're going to look at the F1 generation right here, and we're going to mate those with each other to get the F2 generation. And then we're going to look at the phenotypes in all of these parents and offspring, and we're going to compare the ratios at each generation and see how it compares with what we expect. And I just want to do a quick check-in. Are we still good on the sound and buffering side and everything? Anybody having any issues? I haven't seen comments in a few minutes, so I just want to make sure everything's good. So I'm going to ask. Okay. So tell me if we're okay. And I'm going to keep going. So let's look at the results because they were really interesting. Great, good to hear. Okay, so the observations here were really interesting. So the first observation is the F1 generation right here. They were not intermediate between the parents. They always resembled just one of the parents. The other parent's phenotype, in this case the white flowers, they were absent from the F1. They were just not there, right? The second thing is that the F2 progeny were a mix of the parents. It wasn't just more of the F1 generation. It was a mix of the two parents, including the one that was missing from the F1 generation. So you had white flowers in the P generation, you had no white flowers in the F1, and then the white flowers reappear in the F2. Now, remember when we talked about blending, that finding on its own, incompatible with the blending hypothesis. So that's an interesting finding. The last thing is the numbers. So if you look down here, you have a constant ratio of approximately 3 to 1. So in this case, it was 705 to 224. These are Mendel's actual numbers, by the way. And I'm going to show you more of these numbers in a second. And I just want you to appreciate the dedication it took to do all this by hand with a paintbrush. So we have about 1,000 trials here. And then we follow these observations to other traits, and we find that they were consistent. So if you look right here, here's just a bunch of the traits that Mendel used. So flower color, seed shape, pod color, flower position, and stem length. And you look at these ratios, it's all approximately 3 to 1. 3.15, 3.01, 2.96, 3.14, 2.82, whatever. It's all about 3 to 1. And these numbers are quite large. So you can look here for seed color, 8,000. 8,000 observations because you get a bunch of seeds for each cross. So you get a lot of seeds, right? Each pod contains a bunch of seeds. In this one, it's like 600. Up here, it's about 1,000. Down here, it's a little less than 1,000. Over here, about 1,000, right? So we have big data sets, consistent findings, shows us very clearly that those results I just showed you, these results, not a fluke. This is typically what happens. So F1, you lose one of the phenotypes. F2, the phenotype that you lost comes back. The F1 was not an intermediate. And then in that F2 generation, you got a constant 3 to 1 ratio. So let's bring this home. And we're going short tonight because if I did the next part, it would be way too long. So to conclude, no intermediate phenotypes appeared. The lost phenotypes reappear. Blending cannot explain either of these observations. So our blending hypothesis for inheritance, that is rejected. We have disproved, and by we I mean Mendel, has disproved the blending hypothesis for inheritance. Okay, that's what we have showed right here. So to summarize, because like I said, we're going to keep it short tonight, and then the next one will be a little bit, we'll be expanding on this and probably a little bit longer. Mendel, he used pea plants to determine the rules of inheritance. Coming in, we had two competing hypotheses. We had the blending hypothesis, which is kind of consensus at the time, and then the theory of particulate inheritance, which was Mendel's idea. And he, through the observations we just discussed, disproved the blending hypothesis for inheritance. So what we're going to do next time is talk about Mendel's particulate inheritance model. So we're actually going to get into, okay, today was what did we disprove? Next time it's what replaced it. And we're going to get into the nitty-gritty of that model, including, get pumped everybody, because you know what's going to be in there next week? Punnett squares. All right, so that's it for tonight. Does anybody have any questions about anything we talked about? Or if you feel like you missed anything in the when it was buffering at different times, is there anything anybody is unclear about that I can clarify? I don't see any questions, so I think we're going to call it good. Everybody good? We understand why there's no blending inheritance. Next time we're going to talk about particulate inheritance. It's going to be great. Bring your friends. I am super excited. Who is this? How is this for creationists? This is for creationists because a lot of creationists don't. Let me pull that question on screen. This is for creationists because a lot of creationists don't understand the basics of evolutionary theory. So I'm doing a series to just lay the foundation, and it's adapted from the intro evolution class that I teach. And so what we're doing is laying the foundation before we get into the things like selection and drift and like population genetics and all that stuff. We have to just cover the very basics of starting with Mendelian inheritance. So that's what we're doing here. It'll be a few more weeks before we get to like the actual evolution stuff, but we'll get there. I promise. All right. Okay, well, thanks, everybody. Thank you all for coming. Have a wonderful evening. And one more thing for all 12 of you here and anyone who watches this down the road. If you're watching this before the first week of November and you're in the United States, for God's sake, vote for Joe Biden, please. just do that for me. Thank you. Have a great night, everybody. Bye.